Yeah, so welcome to this biology colloquium. So it's a pleasure to have Professor Supratim Sengupta of the Department of Physics, ISER, Kolkata, giving a talk on one of his many research interests on origin of life. So Professor Sengupta did his PhD from Institute of Physics, Bhubaneswar. And then after that, he did postdocs at University of Alberta, McMaster, and Dalhousie, all in Canada, and joined the faculty of School of Physical Sciences in JNU in 2007. From 2011, he's been in ISER, where he's been working on a large number of interesting topics, including game theory, evolutionary game theory, origin of life, and so on. And we have actually currently two of his master students in our PhD program. So we are all looking forward to the talk. So Shupratim.
Yeah, thank you, Shitabro, for making this visit possible. I'm happy to be here. So I'll be talking about some work that I have been doing for the last 5 years or so with a grad student of mine who just graduated. So about 4 billion years ago, a remarkable series of incidents led to the emergence of the first living organism. So what was this organism like, and what were the processes that led to its emergence? So in order to understand and appreciate the problem of origin of life, it is perhaps helpful to recall what the key features of evolution as we know it are. So as far as evolution as we know it, we know that heritable genetic information is encoded in DNA sequences. We know that phenotypic traits are determined by genes. So there's some sort of a genotype to phenotype mapping, which allows us to look at a gene or a set of genes and in principle predict, for example, the wing patterns of a peppered moth. We also know that random mutations produce variants on which selection acts. And under certain environmental conditions, one of those variants can be selectively advantageous over the others, and it can get selected. For instance, the peppered moth, which is light colored, can get selected when the background bark of the tree is light colored. When the environmental situation changes, this kind of peppered moth gets preferentially selected and increases in number. We also know that evolution is characterized by vertical descent. So individuals pass on their genetic information to their offspring through a process of error-prone replication, and accumulated changes over time lead to speciation, but it also allows us to understand the evolutionary relationship between different organisms and in principle, again, allow us to identify, get to the root of this tree and identify the last universal common ancestor of all living organisms. So the reason I went through this list was to emphasize that about 4 billion years ago, none of these mechanisms were in action, right? So there were no genes to begin with. There were no genes to begin with. There were, of course, no organisms, none of the complex machinery that is necessary for protein synthesis or replication of a genome that happens in every single living cell today existed at that time. So all these processes had to evolve. So in order to understand the origin of life, it is perhaps most important to define what we mean by life. And this was a task given to a group of scientists by a group formed by NASA. And the definition that they came up with is that life is a self-sustained chemical system capable of undergoing Darwinian evolution. Now, this definition is not perfect. There are certain drawbacks associated with it, but it is nevertheless a good working definition to begin with. And the reason for that is that it allows us to break up the problem of origin of life into chunks. So we can ask, for instance, how did a bunch of chemicals come together and start a self-sustained chemical system? And then we can ask, if such a self-sustained chemical system was formed, how did it eventually start undergoing Darwinian evolution? So these two problems are complicated. And so what we have in mind is by starting from a bunch of chemicals, which in this case are nucleotides that make up RNA sequences or DNA sequences, we should be able to produce, long RNA sequences which are functional. That means they can have, for example, certain enzymatic activity. And not only is it enough to produce RNA sequences which are functional, in order to produce a self-sustained chemical system, it is necessary for these kinds of functional biomolecules to be encapsulated in compartments. And these compartments have to be able to grow and divide by a certain mechanism. It can produce a population of such compartments, and then there has to be variation among that population on which selection can act, and a mechanism by which selection can preferably select certain types of compartments over others have to be worked out. So all these steps are absolutely essential to understand how we can produce or how a self-sustained chemical system that is capable of undergoing Darwinian evolution can emerge. Now, one of the hypotheses that was put forward to explain the origin of life is called the RNA world hypothesis. So this hypothesis is based on the fact that in a current world, we have division of labor in the sense that information storage is carried out by DNA sequences, but but enzymatic active catalysis and regulation is carried out primarily by proteins. So the hypothesis said that in a primordial world around 4 billion years ago, RNA performed all these three kinds of activities. It was capable of storing biological information, it was capable of acting as an enzyme, and it was also capable of regulating gene expression when genes arose. And this evidence comes from modern day organisms because we have seen that RNA is capable of, one can produce RNA enzymes called ribozymes, which can act as catalysts. And there are RNA regulators which have been found to regulate gene expression in a large number of organisms. So this led to the hypothesis of the RNA world, where RNA can act as an information storing molecule. It can catalyze chemical reactions and participate in regulation. So, but nevertheless, this hypothesis is a long way from taking us towards an understanding of how the first living organism came into being. So in order to answer that question, we need to break up the problem into parts. So we need to understand how non-enzymatic reactions led to the formation of long RNA polymers, which eventually turned out to be functional. In other words, they could start acting as ribozymes and catalyzing chemical reactions. Once these ribozymes emerged and other RNA molecules emerged, they have to be enclosed in compartments which are typically formed of lipids. So you need not only the essential building blocks, which are RNA nucleotides, you need RNA sequences to be produced. These RNA sequences need to be functional and create enzymes like a ribozyme. And you need lipids to act as compartments which encapsulate these RNA sequences. But once this system is in place, we still need to understand how these protocellular compartments grow and divide and transfer genetic information from the parent to the daughter protocells. So that's the challenge of understanding the problem of origin of life in the context of the RNA world. So here are the key questions. So I'll break up this talk into three parts. In the first part, I'll talk about how just non-enzymatic reactions can lead to formation of RNA sequences which can have complex secondary structures, which can be thought of as a proxy for biological function. And then I'll talk about how these RNA sequences, including ribozymes, can get encapsulated in protocells. And once they are encapsulated in protocells, what's the process by which these protocells can start to grow and divide, and how variation between the protocells in a population can eventually select certain types of protocells over others. And if so, how likely are protocells which contain multiple different types of ribozymes, thereby exhibiting functional diversity in the ribozyme content, proliferated in that population. So, and finally, we need to understand how genetically encoded information is transferred from the parent to the daughter cell. So this requires genetic information to be encoded in DNA sequences. And so in the last part of the talk, I'll give some insight into how the transition from the RNA world to the RNA DNA world might have occurred. Okay, so let's talk about how we can form long polymers, and some of them can become functional using just non-enzymatic reactions. The reason this is a challenge is because non-enzymatic reactions are typically quite slow. And there are essentially two different types of processes through which these non-enzymatic reactions can lead to formation of long RNA polymers. So in the first one, you have nucleotides from the pool just joining together end to end to form a long polymer chain. So this process by which one nucleotide comes and joins with another nucleotide is called concatenation. And this can lead to formation of long RNA sequences. But this concatenation process, the rate is quite low compared to another process, which is called a template-directed replication process. So in this kind of process, we have an existing RNA template, and a complementary copy of that RNA template is made when nucleotides from the pool come and form base pairs with the nucleotides that form the RNA template. So in this case, you can see that the nucleotide U is coming and forming a base pair with A, which is a typical Watson-Crick pairing. And so in this way, this is called the primer. This primer can be extended, and if copying is without any errors, then an exact complementary sequence of the template sequence can be produced. But the copying process is typically error-prone, and this is an absolutely crucial point which I want you to keep in mind. So this kind of error-prone process is absolutely essential for creating a diverse set of RNA sequences which are different from the template. And one aspect of this error-prone replication process is sometimes the errors lead to a cascade of subsequent errors, which again change the nature of the primer sequence, which becomes very different from that of the template. Now, this template-directed replication process occurs at a faster rate than the concatenation process, but it is error-prone, as we have seen here. But this replication process can also occur in the presence of an enzyme, provided such an enzyme exists, and that rate is a lot faster. So here is a movie that is obtained from the Jostak Lab website, where they show how a replicase ribozyme can very rapidly create a copy of the template sequence. And once that copy is created, this double-stranded sequence just comes apart at high enough temperatures, and it produces two different strands, one of which would be a template. So it would not be able to fold into a complex secondary structure. But if the sequence which is copied is not an exact complement of the template, then it can fold into a complex secondary structure and act as a ribozyme if it has a specific structure. So this rate is a lot faster. So this mechanism is a way of producing a large number of copies of an RNA sequence, provided there is a ribozyme which catalyzes this template-directed copy. So I want you to keep in mind, because I'll come back to this process later on. But in addition to these processes which are responsible for increasing the length of a sequence, there's a antagonistic process which is responsible for cutting down sequences into chunks. So these act in the opposite direction. So instead of and this process happens through hydrolysis in a structure-dependent manner, which was shown in this paper. So bonds here which are not paired, unlike bonds in this region, are more prone to be chopped up, which leads to smaller segments of the original RNA sequence. And so these two processes act counter to each other. The concatenation and template-directed primer extension process tends to increase the length of the sequence, which is what we want in order to generate long functional polymers, whereas the hydrolysis process tends to break down sequences into smaller bits. And so it is found that if these two processes occur simultaneously, you don't get long sequences, at least not long enough to produce sequences which are characteristic of biological function. So one way around this problem was suggested by Deemer and collaborators some time ago. And they suggested that environmental cycling can solve this problem of this short sequences being produced as a result of chopping up of long RNA sequences. So they suggested that life could have, and this is a hypothesis, that life could have appeared around the edges of hydrothermal pools. And because in those regions, it's typically mineral-rich regions which can act as catalysts for this RNA synthesis through concatenation and primer extension processes. And so the region around this pool would be subject to environmental cycling. So it would go through dry and wet phases because during the day, these regions around the edge would dry up because of the heat. And again, at night, because of again, condensation or rain, this would become wet. So the key idea is that if it is possible to segregate these two kinds of antagonistic processes, one is the template-directed primer extension and concatenation process, the other is the hydrolysis process, into different environmental regimes, then it is possible to produce longer RNA sequences non-enzymatically. So here's a figure which shows that, which we carried out using a toy model. So this shows that the average length of the sequence is below 10 nucleotides when there is no environmental cycling. So all these two antagonistic processes are occurring simultaneously, whereas if we segregate the template-directed primer extension and concatenation process to occur in the dry phase and hydrolysis to occur in the wet phase, then it leads to sequences whose average length can be much larger than this case. So we carried out simulations by segregating reactions in these three different environmental phases: a morning phase, which is suitable for concatenation reactions to take place; an afternoon or evening phase, which we call the semi-wet phase, where template-directed primer extension processes are favored; and a late night or wet phase, where hydrolysis reactions dominate over these other two processes. So we started off with a pool of monomers and tried to understand how we can produce long RNA polymers and what is the maximum length of the long RNA polymers that can be produced non-enzymatically by changing the duration of the dry, semi-wet, and wet phases. And we found that this kind of segregation into these different, of these reactions into these different phases does indeed help a lot. And you can see the average length going up to about 40 nucleotides, and the maximum length, depending on the duration of the dry and semi-wet and wet phases, can go up as much as 100 nucleotides. And the reason these numbers are important is because the simplest of ribozymes, which are capable of acting as enzymes, are roughly of the length of around 50 to 100 nucleotides. So you need at least lengths of 50 nucleotide long sequences to be produced to have any chance of creating ribozymes non-enzymatically. Right? And so then what we did was to look at the nature of the secondary structures that were produced from our simulations. So we classified all the sequences based on their secondary structures and tried to find secondary structures which are complex in the sense that they were associated with certain known secondary structures of ribozymes that were there. So you can see that these 4 types of secondary structures were produced from our simulations. And so these 2 are too simple to have any enzymatic activity. But this one is a structure of a hammerhead ribozyme, which is responsible for cleaving, self-cleaving. Whereas this is like a tRNA, for those of you who knows what a tRNA structure is. So the conclusion from this part of the work is that long and structurally complex RNA sequences can indeed be synthesized through non-enzymatic reactions, provided we segregate the concatenation, the template-directed primer extension, and the hydrolysis reactions in these different environmental phases so that they cannot act simultaneously and chop off sequences into smaller bits. Yes, yeah.
Can you put some geographical constraints based on this? Because the duration of day and night would depend on where you're on the position on the planet, right?
Well,
like, is that a stretch or is it possible?
We did not get into the details of that aspect, but we did vary the duration of the day-night phases and saw under what sort of conditions you can get long sequences. Whether we can assign a geographical location, I think would be too much of a stretch for these kinds of simple models. But one geographical location which has been hypothesized and which inspired us to look at this problem are these areas around hydrothermal pools, volcanic pools, because they are rich in chemicals. There have been some experiments done which shows that they can produce RNA sequences because these minerals present there act as catalytic substrates for these reactions to happen at a slightly faster rate than what you would expect in other situations. So we are working with the assumption that we are confining the geographical location to those regions, but where exactly on Earth, we are not able to say it.
Just one question. If you have these cycles, is it more likely to filter out strands which are more stable because you're actively disintegrating and forming these strands?
On the other hand, it's more likely to filter out long strands if there are no cycles, because if there are no cycles, then hydrolysis and the concatenation and template-directed primer extension processes are occurring simultaneously. So as soon as a strand increases a little bit, it can get chopped off into small pieces. So by actually segregating the reactions into different phases, it has been shown both experimentally and through our simulations that it is possible to create longer sequences. And not only longer sequences, but sequences which are long enough to fold into complex secondary structures, which have known correspondence with ribozyme structures that we know of.
So what happens if you consider longer durations? Like, I think this is kind of an Earth-like scenario, right? Total number of hours is
24.
But if you increase that, then again it keeps on increasing the length of the polymers in this model. So
we haven't looked. We haven't looked at that. But within this itself, there is variation that you can see. We have varied the duration of the semi-wet and dry phases from 4 to 12. But again, assuming that it's a 24-hour cycle. If it's more than 24 hours, in fact, there's some evidence it's the opposite. It was at that time a shorter day-night cycle. It was less than 24 hours, I believe. Yeah.
So since the cycling is present and like once one synthesis cycle has happened, you will have longer stretches which are now which can fold into secondary or structures.
Mm-hmm.
They will be more resistant to the next hydrolysis step, right? Because as you had shown that unprotected strands are more prone to hydrolysis. So will it gradually select for structured RNA in your model?
Not necessarily, because how long a structure, how long a sequence you produce, typically depends on what is the template that you have. So, so you have to have a mechanism by which you can increase the length of the template gradually.
Yes.
And that is done through the concatenation process. So without the concatenation, you if you take just fixed template lengths, you are never going to have.
We will have more stable RNA, which can act as template in the next cycle. So that's...
Yeah, but the thing is, to act as a template, you don't want a complicated secondary structure. So useful templates are ones which do not have a complicated secondary structure, which do not have too many folds.
I see. That is also inbuilt into the model.
So it is inbuilt into the model, but it's also a fact that, I mean, it's difficult to replicate sequences. So in order for it to act as a template, it should be able to allow for this primer extension on that template. Now, if you have a sequence which has a lot of folds, it becomes difficult to extend the primer because it gets stuck at the folds. So it's for a good template, you don't need a complex secondary structure. You should not have a complex secondary structure. On the other hand, for a good enzyme, you need a complex secondary structure. So there has to be this kind of trade-off.
Fidelity is problem.
And functionally suitable molecules.
Fidelity of RNA polymerization isn't a problem.
Of course, fidelity is absolutely a problem, but it is also an advantage initially. So so none of these sequences can get replicated without any errors. But initially, you need variation in sequences, because if it was replicated without errors, so you would get a sequence which is the exact complement of a template, and which would have very little free energy difference between between that sequence and the template sequence, and it would not fold. So in order for a sequence to be produced that can fold, you don't want a high fidelity process. structure that
So it's
we
just
have.
because of the like it uses less number of nucleotides
Yes.
that it's operating.
Yeah.
And all the other structure. Okay, so that one is little, like
It's slightly more
but
complicated.
these are
It
very
has
rare.
more
The
hairpin
others are much more. Okay.
loops.
Okay.
So the more number of hairpin loops you have, the more complex. So this is a structure which is exactly like that of a tRNA. This is called a cloverleaf structure that is characteristic of a tRNA. And these kinds of structures are produced with about 72 to 90 nucleotides. So this is a structure which is similar to a hammerhead ribozyme, which requires about 50 nucleotides. So the longer a sequence you produce, the more complex a structure you can produce.
Okay.
And this is shown here, but then the long sequences become more rare through these non-enzymatic processes only.
Okay. So it seems to me that as you increase the duration of dry phase, there's more complex structures become much more likely. Do you have an explanation for that?
Yeah, so not just the dry phase, but also the dry phase and the semi-wet phase, right? So if you, so there has to be some kind of trade-off. So dry phase and semi-wet phase are preferred because in these two phases, the concatenation and template-directed primer extension processes are dominant. So on the other hand, you want a shorter wet phase because that's when the hydrolysis reaction breaks up the sequences into small chunks and prevents long sequences from forming. So that's the reason. Okay, so the conclusion of this part of the talk is that ribozymes can indeed be created via non-enzymatic processes under favorable environmental conditions. But we need to move ahead and ask how it's not enough to say that ribozymes can be produced. These ribozymes and other RNA sequences must be encapsulated in compartments, and that requires lipids. And once these processes start occurring in compartments, there has to be a mechanism by which these compartments can grow and divide, leading to a population of such protocellular compartments. And then we have to figure out a mechanism by which certain types of protocells get preferentially selected over others. So there has to be variation within the protocellular population for selection to act on those variants and select certain types of protocellular compartments over others. And whether during that selection process, we get protocells which have a large number of functional ribozymes or not is the key problem that we tackle next. Okay, so the challenge then is to figure out a mechanism, a physical mechanism by which the protocellular growth and division process can be coupled. And such a mechanism was discovered around 2004, 2005 by Chen and Jostak, who showed by putting in a bunch of RNA sequences inside a vesicle that the presence of the RNA sequences inside vesicles exert osmotic pressure on the vesicle membrane and leads to a shape instability which increases the rate at which the surface area of the vesicle membrane grows compared to its volume. And this leads to eventually the fission of the vesicle into two parts. So here's a video which shows this. So the way we modeled it is by assuming that once we took a population of protocells, and in each protocell, we introduced these processes through which RNA sequences could be replicated. And once the number of RNA sequences within these protocells exceeded a certain volume, it divided into two protocells, and the components were randomly distributed between these two daughter protocells. So this was the mechanism of growth and division. And so this is a video which shows how this actually happens. So you can see this RNA strands inside these protocells, which create an osmotic pressure on the membrane, which leads to these instabilities, and the membrane grows and then pinches off at one point with the components randomly distributed between these two daughter cells. Okay. So this was the model that we took. And the question that we asked next is, suppose we have this population of protocells which are growing and dividing in this manner, then can we get to a situation where protocells which contain many different types of functional ribozymes proliferate in comparison to those which do not contain any functional RNA strands? And so this was the scope of the next question that we asked. So the key here is to be able to figure out a mechanism by which the number of RNA strands inside a protocell can be increased. And that can happen only through the process of replication. So if we can figure out how to quickly replicate a large number of RNA sequences, then the larger the number of RNA sequences, the more pressure it is going to create on the vesicle membrane, and the more likely it is to divide into two daughter cells in comparison to those protocells which have fewer RNA strands. So this provides a mechanism by which protocells containing a large number of functional RNA sequences are selected over those containing a small number of RNA sequences. So it's a completely physical mechanism which is at work here. And this was essential in the early phases of evolution in order to create a mechanism by which protocells containing a diverse number of functional molecules could emerge and proliferate. So what we did was to look at a specific mechanism of replication called the
sorry for that. So if the protocells, I mean, having a template RNA is producing a number of some other RNAs which are non-functional, but it's still doing the same thing. I mean, many number of but it's still increasing the volume, right? The RNA.
Right.
Then how can the functional one will be the preferred for a division?
Yeah, so if we can have a functional molecule, say a replicase, which speeds up the process of replication, then the protocell containing a replicase will be able to produce many RNA strands compared to a protocell which does not have a replicase. So a protocell with a replicase will be favored then compared to a protocell without a replicase. Will? They are more likely to divide faster. And so that is the objective, to see whether protocells which contain replicases and other functional biomolecules proliferate. So if they do not proliferate, then this kind of mechanism will not lead to any emergence of complex protocells which are capable of growth and division. So there's a mechanism that we adopted for replication, which is called the rolling circle mechanism. And this was,
So once the protocell has formed and the replication is going on, so it also needs newer nucleotides for the replication to continue. And how does then the nucleotide go through the
so the protocell membrane are typically permeable to nucleotides and small RNA segments.
What is the composition of this? Like what lipids are present, generally taken?
So current cells are made up of phospholipids, but in those times, the hypothesis is they are these lipid bilayers which are produced. Well, I have a video which which is not part of the talk. So can I
Yeah,
come
we
back
can
to you
come-
later on about that?
So they are permeable. That was
Yeah. So they are permeable to individual nucleotides and small RNA segments. They don't allow large RNA segments. So once a long RNA is created inside a protocell, it cannot move out of the protocell. It cannot penetrate the membrane.
Question is like for the structured RNA molecules,
Right.
do they need metal cations or are metal cations helpful in their folding?
Yes, yes.
Okay.
That's a good question. So yes, they do need metal cations. So magnesium and iron are essential for formation of these structured RNA molecules.
Lipid bilayer is also permeable to metal cations?
They are permeable, but they're well, there's a long story behind that.
So
They
we can
also
discuss
end up
after.
degrading the membrane. So there has been workarounds which allow metal cations to move into a protocellular compartment without degrading the protocell membrane. So that has been studied and mainly work from Jostak lab. Yeah. Okay, so we looked at a rolling circle mechanism which was inspired by viruses. And the way this mechanism works is we have a circular template, and there's a primer which binds to the template, and then it gets extended all the way. And then the strand gets displaced as the primer keeps getting extended. And there's a hanging end which eventually breaks up and forms an open-ended RNA strand. And the reason we considered this rolling circle mechanism is because there's a problem that is called the strand separation problem, which occurs for linear templates. So two strands which are produced after replication have to separate, as you saw in that video. But it becomes difficult to separate them for linear strands unless you have very high temperatures, which is again a limiting factor in this case. But for the rolling circle mechanism, that problem is avoided because of the strand displacement, which allows the replication process to continue and these strands to eventually get chopped off, right? So we incorporated this mechanism of replication in each protocell. And initially, there were these processes that were involved. You had a single circular RNA, which can become a double-stranded RNA through this rolling circle replication process. And it could happen both non-enzymatically as well as enzymatically. Then the double-stranded RNA would become an open-ended RNA through this strand displacement mechanism that I showed. And this can also happen both non-enzymatically and enzymatically. And sometimes this open-ended RNA, which is supposed to be an exact copy of the complement of the circular template, but it is not because of the error-prone nature of the replication process. Sometimes it will fold into a complex secondary structure, which can acquire biological function, like that of a replicase ribozyme. So this can, in this way, a replicase ribozyme can be produced just by a chance replication event. And sometimes the open-ended RNA can also join the open ends to form a circular template. So these are the kind of reactions that we modeled, and there was a degradation of all these types of chemical species into their fundamental components. And so we argued that if through this error-prone rolling circle replication process, functional biomolecules such as different types of ribozymes could emerge by chance, then they may have a beneficial effect on the protocell which encapsulated such functional molecules. And then we wanted to look at how the protocell population evolved and whether functional protocells with large number of functional molecules dominated over those which didn't. And so we wrote down a set of equations that were solved numerically inside each protocell in a population of many protocells. And so even though the set looks complicated, the basic idea is you take one chemical species and look at ways in which it can increase its number. And that happens when some other chemical species get converted to that chemical species, and ways in which it can decrease its number, which happens when that chemical species get converted to some other chemical species or degrades completely into its fundamental units. So this is the basic form of the equation that of all the equations that you see, and S, D, and L stands for circular RNA, double-stranded RNA, and open-ended RNA. But during this process of non-enzymatic replication, there is a small, we also introduced a small probability with which functional biomolecules could be produced. So these are enzymes of different types. So PR corresponds to a replicase, which enhances the rate of replication. PC corresponds to a cyclase, which takes an open-ended RNA and folds it into a circular template. And PN and PP are two different types of enzymes, which we also considered. So PN is a nucleotide synthase formation enzyme. In monomer poor environments, this is essential for the replication process to continue in order to produce new sequences. So we looked at this set of equations inside each protocell and asked what is the condition under which protocells with different functional biomolecules could proliferate and dominate the population. And we saw that if you look at just these two different types of ribozyme, the replicase and a cyclase, where the replicase is one which speeds up the replication process, and a cyclase is one which takes a open-ended RNA and makes a circular template out of it, then we saw that the ideal situation is if these two formation probabilities are similar. In that situation, protocells containing both replicase and a cyclase would dominate in the population. You can see that around 75% of the protocells have both replicase and cyclase in them, when these probabilities of their emergence are roughly comparable. On the other hand, if the replicase formation probability is smaller than that of a cyclase, that means there are cyclases which can take a lot of open-ended strands and build circular templates out of them, but there are not enough replicases which can act on those circular templates and create new copies of the strands. Then, of course, the number of protocells which contain both of them are much smaller, right? And so the reason is that if you have a larger replicase formation probability compared to a cyclase formation probability, a lot of replicases will be produced, but replicases need to act on circular templates. So there are not enough circular templates present in the protocell. So the replicases become useless. They cannot act. On the other hand, if the probability of formation of a cyclase is larger, compared to a replicase, then the opposite conundrum is observed. You have a lot of circular templates, but not enough replicases which can act on those circular templates. So the optimal situation is one which corresponds to the case where both formation probabilities are roughly comparable. So we did it for, in the interest of time, I'm going to skip these slides. We did it for a lot of different types of enzymes which could be produced by chance, initially through non-enzymatic replication process, but then subsequently, once a replicase emerges, through enzymatic replication process. And we found that it's possible to look at a hierarchy of emergence of biological complexity which confers a stepwise increase in selective advantage to protocells which contain a larger number of functionally distinct ribozymes. So, yeah, so the conclusion of this part of the talk says that it is possible for protocells containing a diverse set of functional ribozymes to emerge and proliferate and dominate the population, provided they act synergistically with each other, so that their creation probabilities are roughly comparable. So this still leaves open the question of encoding of heritable traits. So in the work that I described so far, none of these sequences were encoded in a gene which could be passed on from the parent to the daughter protocell. But in order to talk about Darwinian evolution and the transition to Darwinian evolution, we need to get to a stage where information is encoded in DNA sequences, which can then be passed on in a heritable manner from the parent to the daughter. And there's experimental evidence which, based on experiments done recently, which led us to address this question. So far we were sticking to the RNA world only. So there were no DNA nucleotides in the picture. But then these set of experiments showed that it is possible for a DNA template along with DNA nucleotide to create a new copy of a DNA sequence. Okay. So in order for heritable information to be encoded, we need DNA. So there has to be a mechanism by which useful information, such as the sequence information of a ribosome, is encoded in a DNA sequence, right? And these experiments suggest it is possible if we introduce DNA into the picture, and then you can have DNA templates using DNA nucleotides to make copies of DNA sequences. You can have an RNA template using DNA nucleotides to make DNA sequences. You can have an RNA template along with a reverse transcriptase ribosome with and DNA nucleotides, which can speed up this process of DNA sequence formation and so on. So this allowed us to suggest that by including DNA monomers into the picture, it is possible to increase the space of reactions and introduce reactions which could produce DNA sequences which can potentially encode functionally useful RNAs. And so this is one set of reaction that we included in the picture, and this is a crucial one, which is an autocatalytic reaction, where you have a DNA template encoding a replicase ribosome, on which a replicase ribosome acts to create a copy of a replicase ribosome itself. So this is an autocatalytic process. So we again modified the set of equations that we could work with inside each protocell to include the effect of DNA sequences and DNA nucleotides into the picture, and looked at a competition between different types of protocells. So in this case, protocells which have a larger number of ribozymes would be preferentially selected compared to those that didn't. And we found that if we carry out evolution in this population of protocells, the protocells that emerge and dominate the population are ones which contain one copy of the DNA sequence that encodes the ribosome, the replicase ribosome, and many copies of the replicase ribosome itself. So this suggests that it acts something like a primitive genome, where you expect that based on our understanding of modern genomes in all living organisms, there's only one genome, but many copies of the proteins that are encoded in the genome can be present in a cell. And you can think of this as a primitive genome with just one gene, which is encoded in the DNA sequence. And through this autocatalytic reaction process, it is possible to get a large number of protocells in the population, which contain not only the gene encoding the replicase ribozyme, but also many copies of the replicase ribozyme itself. And so this is a result which suggests that protocells with replicase encoded DNA in low copy numbers and replicases with large copy numbers can proliferate and dominate the population. Okay, so the conclusion, even though I had to rush through this quickly, is that the emergence of a promiscuous polymerase ribozyme, which can facilitate the transition from an RNA world to an RNA DNA world, is vital for heritable genetic information to be encoded in DNA sequences. And I'll stop by acknowledging the work that has been done primarily by Shubham, who was a graduate student in my group and who just graduated and is going to move to Sweden for his postdoc. Initial part of the work was done with Sudha Rajamani from ICER Pune, who was our experimental collaborator, who provided all the experimental reaction rates for these processes. And Julia, who was a computational physicist, a friend and collaborator with whom we did the initial work. And Neeraja was Sudha's student who did the experiments determining the rates. So thank you for your attention. There was a question here in the chat.
Yeah, can I ask? Okay, that is the maintenance of functional RNA enzymes. A long chain is difficult due to lack of fidelity in RNA polymerization. Actually, there was an experimental result shown around 2020 where they showed that if you start with a small set of ligases in a test tube, then you end up only with a very small functional set. In contrast, whether there was a proposal way back in the 1980s by R. Balasubramaniam of Madras University, who suggested that you can have small functional RNA units, and they showed by conformational analysis that you can use them to actually look at RNA peptide. You can look at peptide interactions and how they can select out peptide amino acids and so on. So, is it possible to have small? They looked at what they call PIT, primitive tRNA, primitive RNA, interacting RNA or something. So they were pentameric, if I remember, they were pentamers of RNA, of nucleotides, ribonucleotides, which they showed could be possible. It was a theoretical study. So is it possible you can look at that aspect?
So the question is whether we can start with small RNA segments and build long RNA sequences by joining together those segments. Is that
no? Just have small RNA segments alone that are functional, and then show that you can build up a system which later on emerges into larger ones. Once there is some fidelity that comes into the system because of the DNA and other process, then we can have longer chains. But RNA as such is not capable of producing longer chains, which are always functional. That's the experimental evidence that was shown.
No, but I mean, so I'm not aware of small RNA sequences being functional. So the smallest RNA sequence which is functional is of the order of 50 nucleotides, which is the hammerhead ribozyme.
Yeah, that is what we have now. I'm just saying that there's a proposal which was actually looked at for some time, but I am not sure what happened experimentally after that. But yeah.
Yeah, so we worked on with the assumption that small RNA sequences are not functional, and in order to get functional RNA sequences, it should be long enough to be able to fold into complex secondary structures. And
Yeah, but we
that's
have
why
small interacting. We know now that we have small bits of RNA that are actually functional. No, there are siRNA and so many other RNA types have come now where there are small bits of RNA that really do the work, much smaller than the hammerhead ribosome and so on.
I mean, it's a bit problematic to compare the kind of RNAs like siRNA with the situations that existed 4 billion years ago, because the small RNAs that exist now are typically work within the context of a modern living cell. And they always work in conjunction with a lot of other functional biomolecules, including proteins and so on. So I am talking about a situation where none of these existed. I mean, there were no molecular machinery which had evolved at that stage.
Yeah, no. The point is small RNA are capable of functionality. It's not necessarily in the context of only now what we see, but if you think of a primordial system where you have only small RNAs which are functional, and then can one build up a full system? That's the thing, because obviously earlier people thought small RNAs are not functional, but now small RNAs are found to be functional, a lot of them. But we think of it now as they could be what is archaeal system, no, things which are evolutionary remnants of what we say, especially since they are mostly from viruses, and viruses are no carriers. So it could be that these are leftovers from the prebiotic stem, which are still being used now in the current day context.
Yeah, well, I'll definitely look up what you are suggesting, but I am currently not aware of small RNAs being functional, but I may be wrong. So I'll have a look, definitely. Yeah, maybe I can, so in response to the question that I put off till the end, I can, about the kind of vesicles that are produced. This is again a video taken from the Jostak lab website. You see these kind of structures. So these are called micelles, which are, and there's a molecule, each of these molecules, they have a hydrophobic end and a hydrophilic end. And the hydrophobic end tends to join with another molecule with the hydrophobic end to avoid the aqueous solution, and the hydrophilic end is the one, and then they can self-assemble into these spherical vesicle-like structures, and it allows for transport from outside to inside and inside to outside of small molecules such as monomers. And sorry. Lipid composition is different from current membrane compositions. Membrane compositions are rich in phospholipids. But at that time, people have worked with fatty acid vesicles and even coacervates. So coacervates are structures which are formed from liquid-liquid phase separation. So they are called membraneless droplets. They don't have a membrane at all.